would you like to record on? It's a real treat to be here today with two very special speakers, Annelies Beery and Irvin Zucker. I'll start by introducing Irv. He got his PhD in 1964 at the University of Chicago, and he has been professor in the Department of Psychology from 1966 until 2009 when he became professor of the graduate school. He's also co-appointed in the Department of Integrative Biology. Irv was president of the Society for Research on Biological Rhythms. He received the Lehrman Lifetime Achievement Award from the Society of Behavioral Neuroendocrinology. And one paper of the many, many papers of Irv's that I'd like to highlight is Stefan and Zucker, 1972, which highlights the discovery of the suprachiasmatic nucleocircadian pacemaker in hypothalamus. And so there's over 2,500 citations, and this is a real landmark in the history of neuroscience that we can all be proud of as occurring at UC Berkeley. So we'll borrow some pride there from Irv's accomplishments. So I think everybody bringing in graduate students to visit next week should be telling them about this discovery. Annelies Beery is assistant professor, just arriving back to us here at the Department of Integrative Biology. She was also professor at Smith College. We were lucky to lure her back. She was at UCSF as a postdoc, as a Robert Wood Johnson Health and Society Scholar. That's a really interesting program that's produced such interesting scientists. And she did her PhD here, finishing in 2008 at UC Berkeley with Irv Zucker. And so one reason to invite both of you back here today is to celebrate this long collaboration and mentoring and collaboration relationship that you've had over these years. Annelies is winner of the 2015 Frank Beach Award from the Society for Behavioral Neuroendocrinology. And one paper I'd like to, some paper titles that I'd like to highlight for Annelies that I think everyone here would be interested in. One of them starts with Stress in Groups, Stress, Social Behavior, and Resilience, Social Selectivity, and Social Maturation or Motivation. And so I believe that Irv and Annelies don't have time to talk about their basic science and work on the brain and behavior in animals, particularly unusual rodent models that they've studied. They're really interesting changes in behavior with season and context. I really urge you to go look at their work. Instead, I think today they're going to talk about this other line of research that's been critically important in changing the landscape of American research and the granting agencies and the focus of research for the whole country. And the title of their talk is Pervasive Neglect of Females in Biomedical Research is a New Day Dawning. And with that, I'll hand it over to Annelies and Irv.
Great. And I'm going to try and move the Zoom around so that we can see people at the same time as the talk. You'll have to let us know. Oh, you disappeared. Can you see us in our main slide?
We see you, but not your slide yet.
Oh, because I need to get over to this screen. There we go. That should work.
We see that. It's
okay, now you should see our main slide.
There it is, yes.
Great.
So
all right.
You should give me feedback. I speak very softly and I don't carry a big stick. So if you can't hear me, please speak up. First of all, I would like to thank whoever was responsible for inviting us. This is the first time that I'm engaging in an academic pas de deux, and I can't think of anybody I'd rather do it with than Annelies. For a couple of the slides, including this first one, I'm going to make a suggestion, which is if you're the kind of person who gathers information more effectively by reading, you could look at the slide. If it's better for you to listen, then don't look at the slide. In general, it's not good to have the two modes competing with each other. And this is just to give you some background that sets the stage for what we're going to be talking about today. So until not too long ago, men were considered representative of Homo sapiens. and any deviations from the male norm were viewed as atypical or abnormal. And until recently, people who were doing research labored under the assumption that the information that they gathered from males could be generalized without modification to females. There's now an abundance of evidence to suggest that that is just simply not true. And the belief that hormonal variations associated with estrus and menstrual cycles render females more variable than males has for a very long time discouraged the inclusion of women and female rodent models in experimental protocols. And this has had a very serious negative impact on the quality of medical care for women. And one of the things that we're going to be doing today is dispelling this myth. And some people have also justified not studying females because they believe that you can only get interpretable data when females are monitored at each of several stages of their estrus or menstrual cycles. In the case of rodents, typically mice and rats, the estrus cycle is 4 days long. And that would mean, if you believe this, that you would have to study them at 4 different stages. And first of all, there are many biologists who don't know how to monitor estrus cycles. And even if you did know and you had to do that, it would be much more expensive. And that's been used as a justification for excluding females.
Great. So as you can already tell, 2 slides in, Irv and I are giving this talk jointly, which is a real treat. And Irv is going to start with a preamble on how he became involved in this topic. And then I'll talk about a project that we undertook together in 2009 after I left his lab, although you never really leave, and a follow-up that I collaborated on in 2019. Together, we're going to discredit 3 different myths or assumptions that get in the way of using females in biological research. And then we'll both talk about studies aimed at addressing sources of variability in males and females. And then Irv will talk about an amazing labor of love that he completed with Brian Prendergast after his official retirement on human sex differences in response to prescription drugs. And then finally, as there's time at the end, if there's time, I'll talk about some recommendations to wrap things up.
So I am going to start with a brief account of how I ended up studying sex differences, which perforce means that you end up studying females. My journey mirrors the more general pattern in evolution of studies of sex as a biological variable. And as you will notice as we go through our presentation, my involvement in this project has, with virtually no exceptions, been in collaboration with Annalise, whom you've seen, and with Brian Prendergast, whose photo is there, but who, to my great pleasure, is now also on this Zoom. Brian has been a professor at the University of Chicago for 20 years. As I go through my journey, you will see that I am not going to be subject to holier than thou a mantle. So my very first study in 1960, my advisor, who was an experienced researcher, advised me to go to the vivarium and obtain 76 male rats. I was very new to research. I certainly never occurred to me to ask him why they were male rats. And from my current perspective, I'm not sure I could have told the difference between a male rat and a female rat. That came to, my ability to do that came about somewhat later. Then, but the fact that I ended up being directed to male rats reflected a then existing strong bias against incorporating females in research. A year later, I did an experiment in which I ended up studying both sexes. I was forced that by the nature of the experiment, but I'll note that I didn't analyze data by sex. As Annalise will talk later on, that's a no-no. Then several years later, when I was doing postdoctoral work, I investigated the role of progesterone in controlling the sex behavior of female guinea pigs. So you could think that I get some points for doing that, but I don't really, because reproductive biologists have never neglected females, and that's what I was doing at the time. Fast forward some 15 years later, and for the very first time, I do a study in collaboration with Donna Bell, in which we study both males and females, and we actually analyzed the data by sex. And this study was an outgrowth of the experience I had as a postdoc, where my advisor, William C. Young, did a landmark study, probably the most important study in behavioral endocrinology, where he and his coworkers showed that exposure to androgens during early stages of development for females would masculinize their external genitalia, would masculinize their male sex behavior, and would defeminize them. So what we did then was extended this line of work to a different species with different endpoints and got similar kinds of results. So I then fast forward several decades to what was a banner year in our lab, 1990, when 3 postdocs and 5 graduate students managed to publish 11 papers. And I wanted to see what the sex distribution was. And it turns out that 5 of the studies were on only females, 3 on only males, 3 on both sexes, and 2 analyzed data by sex. The lead author on all but 2 of the papers was a woman, and of the 3 studies done on males, 2 of them were done by male investigators. So if you look at this, you might end up floating the hypothesis that if you want to have more studies of female animals, oh, and I should mention only 2 of these studies had anything to do with reproduction. If you want to have more studies of female animals, recruit and retain women investigators. Now let's get to how I got involved in sex bias research. This was mostly by accident, but by way of background, what put the bee in my bonnet was in 2001, Sasha Kaufman, at the time was a graduate student in integrative biology, and I submitted an article to the American Journal of Physiology, and it was a study that had absolutely nothing to do with reproduction. It was on the adaptive significance of fur, and it got very good reviews, but one of the reviewers asked us to justify why we were using only females. And I was irked by that comment, and I wrote a very strong rebuttal about being asked to do that, in which I mentioned that scores of studies that my collaborators and I had published using males, nobody had ever asked me to justify the exclusive use of males. So this was yet another form of sexism. Then some 7 years later, I was invited to give the keynote address to a conference on animal behavior at Indiana University. And I decided that the research then going in my lab wasn't going to be of particular interest to that audience. So I thought of some things that I could talk about that they would be interested in. And one of them was to talk to them about how different disciplines use males and females. And I did a 4 or 5 hour survey in the library. I looked at 6 different journals, probably 20 articles from each journal. And I categorized whether they were studying males, females, both sexes, or they didn't tell you what sex they were studying. And I'm just going to look at a couple. Look at the first column on the left. It's the American Journal of Physiology, Regulatory, Integrative, and Comparative Physiology. 80% of the studies are on males, none are on females, none use both sexes, and in 20, they don't even bother to tell you what the sex is. Go over 2 more columns, the Journal of Neuroscience, 30% males, no females, 20% both, and an astonishing 50% don't tell you what it is. And the last column I'll talk about, which Annalise will come back to later, and she'll tell you why we get this distribution. In animal behavior, no studies just on males or just on females, 70% do both, and 30% don't tell you what sex they're studying. At the end of this talk, 2 individuals, Julia Heyman, the director of the Kinsey Institute, and Greg Demas, who was a biology professor at Indiana, come up to me and they say, that's really interesting what you said about sexism. You should expand and write this up. And I thanked them for their interest and said, I had a very busy lab and I didn't have any time to do anything like that. And I got home and thought it was the end of that. And Greg, who, because he was my intellectual grandson, thought he had the license to harass me, kept bugging me to write this article, do this kind of analysis. And finally, he prevailed on me. And then I did what I have been known to do many a time, is enlist a collaborator who's more talented than I am and has greater skills. And I turned to Annalise Beery. Take it away, Annalise.
Didn't do that in our rehearsal. All right. So as Irv mentioned, in late 2009, I became his partner in crime for a more thorough and extensive survey of sex bias in the literature. So I'm going to start by telling you a little bit about how we went about that process. So we came up with a list of 4 journals in each of 10 different biological disciplines, ranging from general biology to neuroscience, field biology, that involved working with animals and/or human subjects. And then for each of those different journals, we examined the first 20 articles of the year 2009 that met our inclusion criteria. So for example, they weren't conducted on cell lines or Drosophila. And then once we found those articles, we went through them in great detail, looking for what species did they use, what sexes did they use, and did they analyze their results by sex. Now, I'll just mention that there's nothing like this exercise to give you strong feelings about where authors should state important details about their study. So some of them mentioned sex in the title. More often, it was in the abstract. Plenty of articles, you have to go down to the methods section to find the sexes that were used. And many more articles, we didn't find it there. And when that was the case, rather than assume that they didn't specify the sexes, we went into the online supplementary materials and dug through all of those, looking for any indication of sex whatsoever. So I'll just highlight that we were both generous in our interpretation. If there were 4 studies and one of them involved females in one part of the study, we still included it as a study that talked about both sexes, and that we were very thorough in making sure that we looked everywhere for the sex data in these studies.
Let me interject, because Annalise probably won't tell you this, that it took hundreds of hours by each of us to get to the bottom of these data.
It was quite an effort. Yeah, so we looked at over 1,600 articles in 42 journals for 2009. We also did some deep inquiry into a couple of different fields in that year. And then we conducted a historical survey in 2 journals that had been published continuously in physiology from 1909 to 2009, each decade along the way, to create a timeline of this. So how bad was the sex bias? Well, I mentioned that we did this for human articles, animal articles. We looked at species lists. Here, I'm just going to show you one of those graphs. And this is actually a reprint that's prettier than the original one that we produced. But this one is for non-human mammals. So if we start by looking just, for example, at the general biology bar, we can see that there's about 20% studies in blue only on males, about 7% in yellow only on females, and another 20% in brown that use both sexes, and then over 50% of the articles that failed to specify sex. So I'll leave you to form your own idea about who you think those studies were on, because we will never know. Since this is the psychology department, zooming over to neuroscience, you can see that there is a strong tendency to have more studies that are male only in blue than female only. That's about 5.5 to 1 male only to female only studies. Still plenty of articles with sex unspecified. As Irv mentioned earlier, reproduction may not get any prizes for this, but they're very good at specifying the sex of whose reproductive system you're studying, and also at studying both females and male reproductive systems. And then when we start to look at behavioral fields, we find that a lot of studies are on both males and females. And that makes sense because a lot of these studies are actually conducted either in the field or in semi-natural enclosures where males and females are always present, as opposed to some of the lab studies where you're choosing to study only males or females. All right, so across all of these fields, we found male bias in 8 out of 10 of them, and reproduction and immunology were the 2 exceptions to that.
Oh, no.
All right. So honing in on neuroscience as an example of sex-based analysis, Here I mentioned earlier, there were 5.5 to 1 male only to female only studies. You can sort of see that blow up in this pie chart here. But when you look at the studies that used both sexes, that green wedge on the right, and you blow that up, it turns out that only 1/5 of those studies analyzed the results by sex. So that when you put it back together into the whole pie and the whole literature, only 5% of studies give us any information about potential similarities and differences between males and females by including them both in studies and then analyzing the results by sex. So we have room to grow. I'll highlight that these findings are consistent with findings from field-specific studies looking at male bias. I think surgical methods might be the worst in this regard, with 80% of studies that specified sex using only males. But it's been found across lots of different fields. In fact, recently a paper showed that even museum collections are strongly male biased. And then across fields, more recently after our paper, a couple of different sort of high throughput approaches have been applied to looking at male bias. For example, text mining PubMed, or an article that recently came out that looked through all the publicly accessible gene expression data. Well, you can infer from the gene expression data, if you have X chromosome data, you can infer the sex of the subjects in both human and mouse studies. And they were able to go back and say, who are the subjects being used? Are they more male or more female? And they found the worst sex bias in neuroscience studies in mice. All right, then in May of 2014, some very exciting plans were announced by the NIH. And I'm just going to excerpt a little quote from this article. They wrote, "The NIH is now developing policies that require applicants to report their plans for the balance of male and female cells and animals in preclinical studies in all future applications, unless sex-specific inclusion is unwarranted based on rigorously defined exceptions." So I think that's important because there are plenty of reasons why you might want to study only females or only males in a particular study, but you just have to give an explanation of why that's the case. And if you don't have a reason for studying only females or only males, then we should include both sexes. And Irv wanted me to point out that 2 of our papers were mentioned in this article as part of the justification for this change in policy.
I would also like to interject that the first time I spoke about these results was in 2010 at 2 talks at the University of Michigan and at Michigan State. And there was no positive response from all the animal researchers. They did not like what we had found because they thought, oh, I'm now going to have to start studying females. And a number of those people were my friends. On the other hand, the people in the audience who studied humans were very receptive to our work.
Mm-hmm. And back in that original sex bias paper, we found that the bias in human clinical trials was much less severe than the bias in animal studies, largely probably in part due in part to the fact that NIH already had regulations guiding the use of women in clinical trials. All right, so after 2014, Nikki Wojtowicz and Teresa Woodruff conducted multiple studies of new NIH panels that were coming along after these guidelines were updated to see how adoption of this policy was faring. And on the 10-year anniversary of the original paper, I worked with them to create a follow-up assessment of sex bias using the same methodology that Irv and I had used in our original efforts. So I'll start with the good news here. You know how to read these graphs from before. Now the colors are blue for male only, red for female only, and purple for both, gray for not declared. And if we look at general biology or across any of these, the good news is that there's a significant increase in studies using both sexes in most of these fields, actually. So if we look at neuroscience again as our focal example, it's a great example of this increase. And the topics that didn't show much increase, for example, like behavior, tended to be the ones that already had a lot of studies on both sexes to begin with. Okay. So this is great news. And I mentioned earlier that we had done a historical timeline in 2 physiology journals. So I'm just going to add those 2 journals to this 2019 update. This is what we had found the first time we'd done the survey, which is mostly a big trend towards having more reporting of which sexes you're using. And after about 1970, having more explanation of sexes, and that's when you start to also see more male-only studies being declared. So when we look from 2009 to 2019, you get an increase in these studies of both sexes and a decrease maybe in male-only studies. Okay, so that's just based on 2 journals, but it gives you a sense of this timeline. There's still room for a lot of improvement, however, in particular, when I mentioned earlier how few studies analyzed the results by sex. You can see the 2009 data in the left bars of each of these different areas of biology, and you can see the 2019 data in the right-hand bars of each of these categories. And the only one that has significant improvement of all of them is pharmacology. And all of the other disciplines had no increase in the percent of articles that were analyzed by sex. And many of them didn't even mention how many females were used. So many studies said we used females and males, and we used 36 animals, but they didn't bother to break down whether they used 2 females and 34 males, or whether they used equal numbers of males and females. So in addition to needing to analyze results by sex, there's a need for specifying in better detail what sexes and sample sizes are used. And then finally, of those articles that did give a rationale, either for use of a single sex or lack of analysis by sex, which was only 4% of all of the articles, Nikki did a text breakdown to see what were the categories that they fell into. And I just want to highlight one, what I think good example, and one of what I think is a bad example for this kind of reasoning. So here's one that says, experiments were done in male mice because the allele is X-linked, and the experiments could not be done in female mice. And there were others where sex wasn't specified because animals were tested on the day of birth before they could be sexed. Here's an example that's not so good. We used males exclusively in these experiments to minimize the possible variability resulting from sex differences. So that leads me to this next area. We're going to start discrediting some myths surrounding the inclusion of females. Irv is going to talk about female variability and the idea that females need to be tested across the estrus cycle. And then I'm going to come in at the end and talk a little bit about sample sizes.
The 2 myths that we're going to be examining are females really more variable than males? And do you need to test females across the different stages of the estrus cycle in order to get meaningful data? The second issue can be addressed by having females whose estrus cycles are not being monitored. So they're at random stages of the cycle. And if it turns out that those animals are no more variable than males, you'll be able to say, I can study males and females without monitoring the estrus cycle. But that'll only be true if you've previously shown that those kinds of females are not more variable than males. So this was a study done with Brian Prendergast and his graduate student, Ken Onishi. And next slide. And what we did here was, first of all, we analyzed almost 10,000 phenotypic traits. And the measure that we used of variability was the coefficient of variation, which is the standard deviation divided by the mean. It's a measure of relative variability. And the traits that were male traits were charted in blue, and the ones that were female traits were charted in red. And then we put both of them on the same graph, which is panel A above. And your eyes are not deceiving you. You're not seeing very much red or blue. What you're seeing is purple. In other words, the 2 distributions almost completely overlap with each other. And then if you look at panel B, we just show some examples in which males are actually more variable than females for studies of metabolism, hormones, and morphology. Next slide. Then we want, using again unstaged females, that means the estrus cycles are not tracked, they're at random stages. We looked to see, were they more variable than males? So the ratio here is females' coefficients of variation divided by female plus male coefficient of variation. And if the value is greater than 0.5, you see that vertical line bisecting the distributions. That means that the variation is female biased, and on the other side, it's male biased. And as you can see, those distributions are very similar to each other. And then if you look at traits that are more than 2 standard deviations from the mean, you find 370 on the left that are male biased and 281 that are female biased. So the basic take home from this is that with unstaged females, they are no more variable than males. So we're still left with trying to understand, why are males just as variable as females? And one of the things that Brian and I thought might be interesting were the conditions under which mice are housed in labs. Typically, a bunch of females are housed in one large cage, and the same thing for a bunch of males in another cage. But what you very quickly discover is the females are pacifists. They don't fight with each other. That's not true with the males. Starting as soon as they're put together, you have an attempt to establish dominance hierarchies, and you have the emergence of an alpha male who ends up basically intimidating the rest of the mice there who are under demonstrable stress. So the idea we had was, maybe that's why males are not anymore, are just as variable as females, because most of them are stressed. So I have to say, just as was true for not specifying the sex that people are studying, most of the studies don't tell you whether the mice are singly or group housed. So we went through and found a sufficient number of studies where we could do this analysis. And if you look at the left panel for males, when this result emerged, I'll speak for myself, I was ecstatic, because as you can see, the group housed males shown in the gray bar are substantially more variable than the singly housed males. I went, well, we have the answer. And then, of course, we made the mistake of analyzing the data for females. And lo and behold, even though the females don't fight, you see exactly the same relation. Singly housed females are less variable than group housed females. And in a striking coincidence, in both sexes, the mean coefficient of variation was 37% lower in single than group housed mice. Next. There is additional evidence that I'm not going to go through with chapter and verse that shows that females are not more variable than males. It's true for gene expression. Brian Prendergast, in collaboration with Jill Becker, did a study similar to the one that he and I had done on mice, and they found exactly the same results in rats, which is another common lab species. And then there were studies in cardiovascular research and many other different traits.
All right, so hopefully at this point in our talk, you are thoroughly convinced that females aren't more variable than males. But unfortunately, this doesn't avoid the concern that if you mix males and females in a study, you're still going to need to double your sample size. And that's something that I heard a lot after these initial papers came out. And I'll just remind you of that quote from the paper that we saw earlier. We used males exclusively in these experiments to minimize the possible variability. They didn't say from using females, they said resulting from sex differences, right? So if you have sex differences between your males and females and you include them both, that's going to increase the variability of your total pool. So around this time, I was writing an article essentially trying to myth bust these different ideas about inclusion of females. And so I put together a little visualization to help us think about how does sex inclusion impact sample sizes. So I'm going to walk you through that visualization, and it essentially consists of a bunch of simulated data and running t-tests and factorial analyses on these different sets of simulated data. So the first scenario that I'm going to show you here is, imagine we have no sex difference between males and females, and we have some effect of a treatment. Come up with your favorite treatment. I've got treatment A and B. That's very boring in here, but whatever your favorite treatment, it has the same effect on males and females. So we have the females on the left, the males on the right. You can see that they're exactly the same. And for all of these examples, I artificially defined the effect size as 1 in order to randomly generate my data around a particular mean and variability. All right, second scenario, let's imagine that we have a large sex difference. And again, there's going to be an effect of the treatment with effect size 1, and there's no interaction between the treatment and the sex in this scenario. So you can see the females have lower overall means than the males, but they both have that same effect of treatment. Okay, and then here's our third scenario, our nightmare scenario, where we have opposing effects on each sex. So there's a strong effect of the treatment that we would have identified if we had studied only males or only females. But if we mush all our males and females together, we're going to get really confused because they have opposite effects in the two different sexes. So here I'm just visualizing for you what happens when you do different kinds of analyses on these data and you run them in simulation. So sometimes you won't find that a result is significantly different, no matter how good that effect size of 1 is. But most of the time, for example, in that red distribution on the left, you'll find that that one comes from sampling 12 females or 12 males. I think I lost the label that comes on this. But here we either take 12 females in that left bar, 12 males in the green bar, or a mixed group of 6 males and 6 females in that blue bar. And here, because there's no sex difference, we have no impact whatsoever of mixing males and females half and half in our study. And if we run a two-way ANOVA with sex and treatment as factors, we also have no effect of mixing males and females in our study, and there's no interaction between sex and treatment because the sexes were the same. Okay, so that's our simple example where obviously you can pool males and females because there are no sex differences at all. Okay, there's the label that says how many are in each group. In the second one of these, now we have a big sex difference. So if we study our 12 females, we get our effect. If we study our 12 males, we get our effect. And if we mix our 6 males and 6 females together, unfortunately, all of our signal doesn't disappear entirely. But in this red bar, you can see that we don't have that heavy weighting towards low and significant p-values. So mixing, not so good if you just mix naively. But when you do your factorial analysis and look at the effect of sex and treatment in a two-way ANOVA, you see you get exactly the same results as you do in a t-test. So there's no loss to combining males and females in a single study when there's no interaction between sex and the treatment variable, even when you have a big sex difference. And you can see that lack of interaction in the bar on the far right. All right, what about our dread scenario? Well, in this case, again, studying 12 males or 12 females will give you the same distribution of significance. They'll give you opposite answers, so you better not extrapolate from one sex to the other, but you could get the result in either sex. Mixing 6 males and 6 females obliterates your significant result, but so does the two-way ANOVA. So the two-way ANOVA can't rescue you in this circumstance. It's not the fix-all, because there was a very strong interaction between sex and treatment. But you can see that in this bar on the right, because the interaction is very significant. So this is the flag that tells you this is the study where you need to perform it in enough females and enough males to study them both separately, because you had a strong effect of sex. So it's not the case that you never have to increase sample size, but only in this third set of circumstances where there's a strong effect, or rather a strong interaction between treatment and sex, where you need to increase the sample size. And in the extreme, it would be doubling, but it doesn't have to be doubling. So a year after that paper, this paper on the bottom right came out actually simulating how much bigger does that sample size have to be. And they concluded that for exploratory studies that were not focused on confirming a sex difference, you could do this with either no increase or sample size or a very small increase in animal number, much like we showed in the previous slide. But if it's a confirmatory study, you think there's sex differences, you need to account for these things, then in general, it required 33% more animals per study. And actually, not on average, that was sort of the worst case scenario. Now, that's if you don't care about the sex differences in and of themselves. So I want to add a very important caveat, that's unless the goal of your study is to understand the sex differences, in which case you might actually need to double the sample sizes in order to fully study the phenomenon separately in males and females. So sometimes that is a very important goal of the study. Okay, so now Irv is going to talk to you about some sources of variability in males and females.
I left you previously with the idea that we had no idea what the source of variability in males is. And it occurred to me that I did not know of a single study in mice or rats in which males were tested on 4 consecutive days at the same time as females were studied on 4 days while they were undergoing estrus cycles. And it was conceivable that for reasons that we don't know, that across days, males would show significant variability. If this were confirmed, it would account for the absence of sex differences in variability. Next slide. At this point, I was approached by Ben Smar, who was a postdoctoral fellow in Lance Kriegfeld's lab. He said he was interested in studying sex differences in and sex bias. So he implanted recording devices into male and female mice that were measuring locomotor activity and body temperature at 1-minute intervals across the day and night. The night is shown by the dark bars and look at the left panel. This is on females. If you look at the leftmost panel at the top, you'll see that there's a very extensive amount of activity almost throughout the night. If you look on the next 3 days, that activity is absent and you see a very sharp decline. If you have your eyes moved over to males, it looks exactly the same for, it looks exactly the same for males on all 4 days as it looks for the females on the 3 days when they're not in estrus. I'm not going to go through pointing out that exactly the same relation holds if you look at core body temperature, which are the graphs on the bottom. So this establishes that there is increased variability in females across days, which we could have told you that starting in the 1920s when people first started measuring locomotor activity in male and female rats, but this confirms it for a number of different measures. And as Annalise pointed out, with female variability, you still don't need to have very great increases in sample size. But Ben went ahead and also looked at variability not across days, but within a day. And that's shown in the panels to the right. And I'll just look at this one, the one that is boxed. And the take-home message is that the daily range and variability is significantly greater in males than it is in females. And that's true if you include all days, which includes the day of estrus, or without estrus in the next panel to the right. So the take-home message from this is that, yes, females are more variable than males across days. Males are more variable within days. So if you're doing an experiment where you're getting measured over the course of a whole day, you're going to have better luck if you're studying females than if you're studying males. And in aggregate, it would appear that these two sources of variability counteract each other or they cancel each other out. And as a result, males and females are no more variable than each other.
All right, so shortly thereafterward, and again in collaboration with Irv, who was interested in this topic at the time, we conducted a similar study in my lab at Smith College to examine whether this was the case for multiple traits in the same way as it was for body temperature. So we looked at how inter or between individual variability compared to within individual variability between the sexes for multiple traits. And then we also looked at whether removing ovarian steroids would reduce variability in females or not. So for this group, although if you've heard me talk about rodents before, it's usually voles or another untypical laboratory model. For this study, we used hamsters because they have a perfect 4-day estrus cycle that's externally monitorable. And so we knew exactly what phase of the cycle these females were in at all times. So we studied intact females overactimized females, and intact males. And then we tracked 3 different behavioral traits over 8 consecutive days in series. After habituation days, we do 8 days of monitoring of the trait. And then we looked at 2 physiological measures. And together, this actually gave us 8 independent traits to study. And each of those traits were studied over 8 consecutive days, which is exactly 2 estrus cycles in a Syrian hamster. So this was actually the undergraduate thesis work of Katie Mashofsky and many helpers. And Katie is currently teaching high school biology in Richmond, California. So she's out here. All right, so how do we read these graphs? This is a temperature graph, and you can see the males are the green bar in the center, and the 2 sets of crossbars are variability within individual on the x-axis and across individuals in the study on the y-axis. So within individuals, it's across days, and across individuals, it's across the group. So males in this particular instance are higher variability in both dimensions than females in this particular study. And the females, you can see the overactimized and intact females are sitting right on top of each other in the top right side of the graph. But we did this for all 8 traits and then pooled them to assess whether there was any particular uniform pattern of variability that came out of this. And I'll give you the short answer, which is there were no significant differences in variability within individuals or across individuals that spanned across traits. So there was essentially trait specificity in who was more variable in each particular way for each particular trait in this study. So you can see that in this density plot in the left-hand graph, we're looking at within individual differences, and again, the coefficient of variation. And the more left-leaning a group is, the more the coefficients of variation sort of pile up on the small side. So if anything, the males were more variable than the females within individual. And if anything, in panel B, the males were less variable across individuals, but that was across a lot of traits, and there were specific traits for which exactly the opposite phenomenon was found. And again, there were no significant differences when we pooled all of these traits together. So that was one of the findings from this study. The 2 other things that came out of it, one is that we got no improvement in variability for females by staging by estrus cycle phase. So that's kind of interesting. We take these females who are cycling naturally, we line them up by which day of the estrus cycle they're in, and they don't have a decrease in variability. So that's more strong evidence that the estrus cycle is not contributing to substantial amounts of variability in females. And then we also found that the overactimized females who don't have cycles had no significant reduction in variability. And I will say that we tested one other trait, which was behavioral estrus, when you put a male in the container with the female and she goes into lordosis, and that one did vary significantly by estrus cycle phase. So just as a sort of a positive proof of concept, of course, some traits do vary substantially by estrus cycle phase, but even though we'd selected some things like sucrose preference where you can find these subtle differences, it didn't improve variability in our study.
I would like you to just take a minute to study this slide, because on the left panel, you see various syndromes, disease states that occur more frequently in women. Some of them are obvious: breast cancer, various endocrine disorders, MS, and then there are health conditions that are different in men and women. The first one, cardiac disease. I think most people know that the symptoms of a coronary infarction, heart attack, are quite different in most cases in a man and in a woman. Same thing is true for migraine headaches, for emboli, for psychiatric disorders. Next slide. I'm just recapitulating something that, actually, I'm saying it for the first time, excuse me. It's been known for several decades that women are at significantly higher risk for adverse drug reactions, nearly a 2-fold greater risk for ADRs than men, and they're more likely to be hospitalized following an ADR. You might ask, what are ADRs? They are things like nausea, depression, anxiety, diarrhea, headaches, more common in women than men. And women also handle drugs differently than men as a result of metabolic enzyme differences, clearance rates through the kidney, and many other physiological differences between the sexes. Next slide. I want to talk to you about a very well-known case study, which is the drug zolpidem, which you may know by the name Ambien. It is a hypnotic used for sleep, and it was approved by the FDA in 1992. And at the time of the approval, it was already known that when men and women are given the same dose of Ambien, the concentration of the drug is 45% higher in women than it is in men. And it was subsequently found that clearance of the drug is 35% slower in women than it is in men. So the drug is around for longer. Despite these striking differences, the FDA did not recommend any reduced doses for women. After decades of post-marketing reports, and it's worth mentioning that most of what we know about the adverse drug reactions comes from post-marketing reports because the pre-approval studies are not of significant duration to show up most of these negative consequences. What was found that women had more cognitive deficits than men, and of particular concern was the fact that after taking Ambien one evening, the next morning, women were involved in significantly more traffic accidents. Well, I found this, so the FDA in around 2007, which is 15 years after the drug is put into general use, they recommend that women should take half the dose that men do. I was quite struck by this, and I then did a search to find out how many other drugs are there where the FDA recommends that women should take lower doses. I could have saved myself a lot of wasted time. There were virtually no such drugs, less than a handful. So then I had the idea, and in collaboration with Brian Prendergast, asked the question, could it be that the sex differences in pharmacokinetics, that is the higher concentrations, slower clearances, are responsible for the increases in adverse drug reactions? So we spent a lot of time looking at a large number of studies. And the title of the article is shown up there. And it turns out that after looking at many hundreds of studies, we only found 86 FDA-approved drugs in which there were known sex differences in pharmacokinetics. For most drugs, they don't have such data. And in order to test our hypothesis, we of course needed to have information about adverse drug reactions. Of those 86 drugs, only 59 of them also told you what the adverse drug reactions are. And then what we found was for the sex differences of the drugs that had a sex difference, greater exposure was present in 88% of these drugs for women. So when you find a sex difference in pharmacokinetics, it is much, much more likely that it is women who are going to have higher concentrations of the drugs. And then what was most interesting to us was among the 52 drugs where we also had data on adverse drug reactions, 50 of the 52 drugs where women had higher pharmacokinetics also had ADRs. There was a concordance of 96% between PK, pharmacokinetics, and adverse drug reactions. In contrast, only 2 of the 7 drugs with male-biased PKs exhibited male-biased adverse drug reaction, a 29% concordance. So to wrap up my part of this presentation, I need to remind you that most drugs currently in use were approved based on clinical trials conducted on men. The absence of sex-stratified pharmacokinetic information in public records for many hundreds of drugs raises the concern that sex differences in pharmacokinetic values are widespread and of clinical significance. Prescribing equal drug doses to men and women neglects sex differences in pharmacokinetics, dimorphisms in body weight, change in many morphological and physiological parameters, and it risks over-medication of women, and in our view, contributes to female bias adverse drug reactions. So we ended up recommending evidence-based dose reductions for women to counteract this sex bias. On average, you should try and find the lowest dose that produces the desired effect and only increase it in women if that doesn't transpire. Annalise?
Well, I'll just add to that, that when this paper came out and I got to see it in an earlier draft as well, I assigned it to a group of students in my seminar at Smith College, and the reaction, they didn't have to read the whole thing. It's encyclopedic in its nature, but focusing on particular drugs and how they had been studied or not. And it was shocking to all of them how little we know about the drugs that we are constantly taking, and how much of it is not in service of women's health in particular. So I consider that paper a great service in many ways. So one recommendation came out of that you just heard. Throughout the talk, you've heard a number of recommendations. So on the research front, include females in your research, either study both sexes or study them separately if you need to, or have a reason for studying only the sex that you study, but don't just exclude females, of course. In terms of medications, you just heard that we need to adjust dosing recommendations for women. And as a female patient, I'm going to take the lowest effective dose that I can find. And then on the reporting front, in addition to reporting sexes and their sample sizes, ideally early in an article, it's also important to analyze results with sex as a factor in the analysis. And that's where I just want to spend the last 2 slides focusing on how should we report on sex differences and how should we analyze sex differences in our data. And as we go into that, I just want to acknowledge that it's not a totally straightforward question, right? So there are a couple of pitfalls that we could worry about. For one thing, what if we make too big of a deal out of sex differences? So there are, in fact, consequences that can be negative to reporting too strongly on sex differences. We can make it seem as if males and females are almost different species from each other, right? But a lot of the time, the sex differences that we're reporting on have a high degree of overlap, and there's a very tiny difference that is nonetheless significant between males and females. So we need to be careful about describing accurately the sex differences that we find and contextualizing them appropriately. On the other side, even overlapping distributions with very slight differences in means can also indicate sex differences in underlying mechanisms. And it's really important to detect those kinds of sex differences. They can have very important clinical applications as well as understanding of fundamental biological mechanisms. So here, the paper that I'm flashing up has to do with pain processing, where there are strong sex differences, not in overall phenotype of pain sensitivity. That varies very significantly, but very smallly, that's not a word, between the sexes, not very much between the sexes, but there are sex-specific mechanisms involving microglia in males and melanocortin receptors in females that are unique and don't cross over. So if we target glial cells, we're targeting male biology in terms of pain pathway and remediation. And if we target melanocortin receptors, well, we haven't done that at a clinical level because the preclinical trials were done mostly on males. And so we don't tend to get there. All right. A third pitfall is highlighted by one of the only papers, I've only found 2, that was critical in print of the NIH policy. And this article came out saying focusing on preclinical sex differences isn't going to address women and men's health disparities. And the point of this article is largely that all of the work that we do on sex differences in rodents can translate to sex differences in humans, but that gender differences in humans and the cultural baggage of being a female or a male, and not just baggage, but all of the significance of it plays a very large role in human health. And I would suggest that this is not an either or scenario. It's not that one is important and that the other isn't, but rather that these are both things that we need to keep track of. All right, so there are a number of guides out there now about how to deal with sex differences in data. I'm highlighting a figure. You don't need to look at all of the detail in it, from a particular article collaboration with Adrian Beltz and Jill Becker. And the point that I do want to pull out is that in most circumstances, the answer to how do you do this kind of analysis, you simply include sex as a variable of interest in your analysis. And if you get differences, or if you don't get differences, describe them. So examine them, look for them in the first place, and then describe them when you get them. What was the effect size? What are the mean differences? And Donna Maney has a very nice visualization of this here. There's always a push these days towards display more of your original data. When you do that, and when you show the distributions and the effect sizes, you can see very nicely how big of an effect this is. And there are many additional guides out there. These are not the only ones. So that's where we wanted to end, and hopefully there's time for questions. And thank you all for coming to this talk, and thank you for the invitation to give the talk. This was really fun.
Indeed.
Thank you so much, both of you. Well, I would like you to maybe close so I can see everybody, and we're open for questions. People would like to raise their hands. Jason's first hand up.
Oh my gosh, this presentation was amazing. I'm so glad I was here for this. I'm so glad that you're doing this work and have been doing this work. Incredible. And you didn't talk too slowly or softly. You talked just right.
Thank you.
I have so many thoughts, but I don't want to. I love when you get. I love when in these talks when you get a variety of questions and things like that. So just really simple, and it's similar to some of where you left off. And so I'm Jason O'Connor. I'm in our social area, and I say that because on the social side of things, there are a lot of arguments about gender versus sex. There are a lot of arguments about what sense to make of it. And so two things I wanted to ask is, one, across everything, I think I know the answer, but across everything you just showed us, would you expect there to be a difference if it was not sex, but rather gender? Right? I don't even know how to ask that question, but it's like the social implications of gender versus the biological implications of sex, and just like the findings that you have in my mind, I'm like, okay, so if we could make it clear, if we could start to, organize these things, these are things that have to do with sex, but I don't know, are these things that may come with how one is received based upon how one presents oneself or how others interpret gender or things of that nature. I'm sorry, I'm just flooding over. That was so much information. It was so awesome. And then the other question is, I guess, more practical from a research standpoint. So I've been introduced to the idea that when it comes to like understanding gender or sex differences, it's best to ask participants something like male, female, other. And if others specify or explain, or creating some type of spectrum type measure. Do you have any thoughts or ideas or feedback on that? That's all. But overall, thank you.
Okay, I'm going to jump in with the first part of that and say that when we're studying animals, we have the advantage of not worrying about gender, right? We talk only about biological sex. So when we're studying animals, we're really just looking at sex differences. And many sex differences do translate to human sex differences, for example, in the cardiovascular system, all sorts of different systems. So we can say that there are sex differences in animals, there are sex differences in humans, and it becomes very difficult, as you said, to start to parse them. Some obvious examples that people have studied of effects of gender on medical treatment in humans include, for example, what's your gender when you ask for a pain medication prescription, or any different social variable, right? So clearly there are ways in which gender influences female health and male health. Clearly there are ways in which there are basic fundamental biological sex differences. And then I think always, it's like almost like nature nurture debates. You can't separate sex and gender. You grow up with both, and sometimes they transition as well. And then it gets even more complicated. That gets into your other question about, how do we assess people's gender, right? But I think to to me, there's no clear benefit in trying to isolate, is this exclusively a sex effect or a gender effect or some interaction between them? I think they're both at play and we should just acknowledge that. So that's just my 2 cents.
I have nothing to add to that.
Anne.
With Jason, I'll just say, wow, what a great presentation. Thank you so much for sharing this work with us. It's so important. And I wanted to ask a question about the variability findings, which I found to be fascinating. And I hope that in the fields that you both work in, other people found them fascinating as well. I think you've definitely put to bed the myth that the reason for not studying females is because there's more variability. You've studied that in so many different interesting ways. I just found that, really compelling. And but I wonder if in your fields, if people also found it compelling that there was so much variability, no matter what the sex is, and how you think about that when studying animals. I may be wrong about this, but I think, in some corners of the field, people assume, well, we'll just, like back when you started with your first example, or get 76 male rats for your study, and that they're all going to be the same because they're rats after all. But there's a tremendous amount of variability there. And so how do was that a big splash when you published these findings as well? Just the whole concept of the great variability that is in these species, regardless of sex?
I'll try and respond to that, Anne. I think that our work has served as a catalyst, and that the much more important thing is what people at the NIH did, because the way you get people to change long ingrained habits is to connect it to something they really want, which in this case is filthy lucre, money. So when the NIH said, if you want to get funded, you have to study females or have a strong rationale for not including them, that woke people up, and you saw the change in the follow-up that Annalise did. So I think that our work is very heavily cited. I don't know how many people actually read it, but they cite it. And I think that the publicity that it got in the popular press and the changes mandated by the NIH are what is important in redressing this problem, not the research that we did. It was just a vehicle to get this thing started. And of course, we're gratified by what happened.
Yeah, I'll add to that on the variability front. I think the conclusions from the studies of animals housed in groups versus singly, right? None of us want to immediately start housing our social animals singly, but it was really interesting to me to see the scale difference of variability from different sources and what a big source of variability that is. I'll also just say from my experience of running that experiment where we tested the same trait over 8 consecutive days and looked at the variability, of course, within individuals and across individuals, just seeing the amount of variability day to day in some of these traits, some of them were highly consistent and some of them were quite variable day to day. So when we take a measure, maybe that's something else we should be thinking about in our research, is for some measures, maybe we should be taking them repeatedly just because of that variability. Although I'll note that one of the tests that we used was a novel object preference test. And we had 8 different novel objects that the animals got across those 8 days. And I was very, very pleased that in males, females, and overactimized females, you could see exactly how good each object was, because all of the groups relatively sniffed that little orange cone more than the standard object versus, some other object. So there was enormous consistency in some of these traits, even behavioral traits across groups. So we didn't just see variability. Yeah, Daniela.
Hi, this was amazing and fantastic to hear you both, fantastic to think about this work this way. A thought that I had about the group housed versus single housed and the variability there, and yes, as you said, that's very alarming to somebody that studies, is, so what about stress, right? Because single housed animals, they're stressed, and I think that pushes all of the variables into, may or may not, but you're seeing an effect of stress there as well. just by singly housing them. And then of course you have variability in the groups of where they are in the social hierarchy.
Yeah, my first thought to that is, wouldn't it be interesting to take those data and separate them out by whether the species were social, right? Because a hamster is actually hunky-dory on its own in a cage and might not, do as well in a co-housed circumstance.
Yeah,
And
but
see if they reduce
those
variability
were all
too.
mice.
Those were all mice.
Yeah. And you can infer from the variability data anyway that the singly housed animals they probably were somewhat stressed, but I think they were less stressed than the ones that were living in groups, especially for the males. You can observe them fighting in the males, and you don't see that in the females.
Could I offer a comment on that, Irv? And Annalise?
I can't hear
Yeah,
you.
Brian, go for it.
And it was to perhaps address Daniela's question. When we did a similar analysis on rats in collaboration with Jill Becker, we didn't find anywhere near the same effect of group housing on variability. In fact, there was mostly no effect of social housing on variability in rats. Again, no sex difference. And so maybe there is something about rats being more inclined towards living in social groups.
Interesting.
Sylvia is next.
Yeah, thank you so much for that wonderful talk. Irv, I've heard you talk about it a few times now, and I'm just really thrilled with the work. And Annalise, welcome to Berkeley. Most of us haven't had a chance to meet you in person yet. So thanks for coming and speaking in psychology. So Irv said something about most clinical drugs that are currently in use not having been tested in females or been reported in females. But then you had earlier said that the NIH guidelines had changed a while back for clinical research in humans. Is that right? And so is it just that most of the drugs currently in use were tested and marketed just a long time ago?
Yes, the most significant event for increasing participation of women in drug tests came from the NIH Revitalization Act of 1993. And in our article, we actually plotted over the course of, you see a big change from the 1990s in the next 10 or 15 years with more women being included. So, and you're absolutely right, Sylvia, that it's because a very high percentage of the drugs were approved prior to 1993. And I'll also mention that there are two categories of women for whom there are very, very few drug tests, and that's pregnant women given drugs and lactating women given drugs. And I actually wrote two separate articles on that, showing that there's been and the impact of a woman getting a drug, say, during pregnancy is frequently not shown in her offspring till 15 or 20 years later, which is not something that you know about at the time that the drug is approved. And I actually found that if you study animal models of some of the drugs that are given, let's say, to counter depression postpartum, you can make very strong predictions as to what the deficits are going to be. And the sad thing is that virtually all of the animal model research is done after the drug has been approved and is being used in women. So, yeah.
That's Rona, then Frederick.
Thank you. I just figured out how to put up my hand in this, so I'm glad. This was a marvelous presentation, and I've heard bits and pieces and read the articles along the way. And what I want to compliment you on is the whole paradigm you've used here to examine these questions and undercut the myths systematically, to test alternate explanations, hypotheses, to present this data to change policy, and then to look at whether it's had an effect in an historical analysis. So it's an amazingly powerful paradigm as a system of studies. And it also opens up the whole important questions that we rarely talk about of who is in our sample, in what proportion, in what social condition are they housed, the interplay of biological and social factors, and then the duration, duration of follow-up and the particular kinds of outcomes you look at. So if you look at your data, Irv and Annalise, some of those outcomes are huge in society. So here we're talking in general, but some of the outcomes for women and for children and outcomes are huge ones. So in a way, this work opens up the possibility for many more contextually and ecologically valid studies and for a different future for psychological science. So thank you for doing this work.
Thank you, Rona. And as you know, I've appreciated your input over the years in getting me to overcome my reluctance to talk to the media, etc. Even though I felt you were browbeating me at times, it was in a good cause. I will also mention that for the last year and a half, I'm pretty much done with sex bias things. My friend and colleague here is much better positioned than I am to carry this forward, as she's already shown in her analyses with other scientists. What I've been doing for the last year and a half is a very similar approach, asking the question of the influence of race, ethnicity on responsiveness to prescription drugs. And there are striking differences according to those dimensions, and it'll probably be another year and a half till any kind of a report emerges. And I'm about to be joined in this enterprise by one Brian Prendergast, who, having finished some other work, is going to be available to work with me on this. So, and that will be my sayonara project.
Frederick.
Yeah, thanks for that talk. Just really wonderful. And it's so nice to see you, Irv, even though I wish it was in person, so it could be even more real. But it's nice to see you with a little posture stamp like this. I have, like, Rona, what you said just was really along the lines of what I was thinking, but more in my small world of using animals in the lab. And I think, Rona, you mentioned this, and Jason kind of indirectly too. I mean, when you start to take into account, not sex differences, but just sex, and I think it brings the issue of, like, males and females, animals live together, and that when you house these animals in separate male cages and separate female cages, they are in a very weird situation that is unnatural. And so I was wondering if I know that, Irv, you might be more interested in the kind of clinical applications, but if this is something that maybe Annalise is thinking about too, in terms of, like, not only having studies where you look at differences between males and females, but also where you have housing situations which are more normal. And what the effect of that would be in terms of, decreasing the stress, for example, and changing the physiology of the animals, that would actually also change your biological results. Clearly changes the behavior. So,
Okay.
kind of an open-ended question, but there it is.
It's almost as if someone fed you that line. So that's why I wanted to jump in here. I'm absolutely interested in what happens in more realistic or semi-naturalistic conditions. And I would say that a big thrust of what we're doing in our lab right now is housing animals together in groups and monitoring their activity and their movements in a more free-form way outside the realm, still using these sort of standard behavioral assays, but also putting them in more naturalistic groups. So you couldn't have asked a better question on that front. I'll say that I have a colleague, James Curley, who studies social hierarchies in mice, and he'll put 16 mice in a big group in an apparatus and study individual, like a lot of detailed behaviors about the formation of the dominance hierarchy in these big groups, but they're always all male or all female. And so I think it's really important that we consider things like what you said about sort of what is the nature of the groups that animals are in and housing animals in actual groups. It's especially important because I study group living, and I'm embarrassed to say that for the past decade, I've done it on singletons and pairs, right? So for us, moving up to groups is a big thing. You want to add?
Frederick, I was very concerned about that issue when I had a lab, and particularly because as a chronobiologist and with animals being kept on light dark cycles and working mostly with nocturnal animals, it was absolutely crazy that for, let's say, the animals on 12 hours light, 12 hours dark cycle, that those animals in nature never, ever spend 12 hours in the light. They'd get killed. So what we did was we introduced into the cage of each animal an opaque small beer jar. And if you came in during the light phase, where do you think the animal would be? In the jar. Mm-hmm. And they would come out very intermittently to try and get some food or to take a drink. And it changed their sleep patterns and many other aspects of behavior. So the context in which we keep the animal is important. And then there was a change in the Office of Lab Animal Care. And the new director said, get rid of the beer bottles. And we got rid of the beer bottles. It was out of our control. So context in which an animal is housed in relation to the way it lives in nature is critical.
These days they do let us we have these PVC tubes we put in the cage. I
In
spent,
Berkeley.
yeah.
All right.
Over the weekend, I cut a bunch of them up on the miter saw to make these long pipes into short
There is
pipes.
progress.
All right. Thank you so much, Annalise and Irv. This has been really a treat. It's really just stunning to see this work you've done. And I wish we had another hour because I would love to start asking questions about the other biological variables that we might have missed and your work on circadian and seasonal biology and the really radical changes in social interactions that occur seasonally is something that I poke everyone else here to go and ask these two about, because it's really fascinating that how context and these biological variables are changing animal behavior. And we're very grateful to see you and want to celebrate your long partnership, men and women working together. It's another good thing to see happening.
So I want to say something. I want to thank all of you for coming and sitting through this data blitz. And it has been a pleasure to be here. And it is a gift that keeps on giving, the number of fantastic students who come through this university and supportive colleagues.
I'll let you have the last word, Herb.
Thank you.
Okay.